wire, and your fingers are actually pointing in the direction where the magnetic field will be flowing. And if you look at the source, these are again our little dipoles formed by our pyramidal cells. If the current is flowing, and as we explained before, is the current flowing in the dendritic tree and the cytoplasm, then we get a magnetic field that is orthogonal to that, and with the direction indicated as just explained by Oersted's trick. And yeah, you have here like the bottom a telephone line here. Of course, in telephone line have electrical current flowing, and then you see the circles indicate the magnetic field, and this is paralleled here by a pyramidal cell on the bottom, where you also see these flows. And yeah, this can so of course the current should be running parallel to the scalp, a bit as shown in the figure on the right. And so that the magnetic field can exit the skull and be picked up. So it means if the current is perpendicular to the scalp, as in the case of these pyramidal cells that we refer to in the case of EEG, then these magnetic fields will stay in the brain and cannot be measured by these magnetic sensors. So in a nutshell, I'm on slide 26 now, that MEG is going to be more sensitive to the activity of pyramidal cells in the walls of the sulci. On the figure right, you see a sulcus, and then of course the pyramidal cells aligned in that sulcus will be picked up, but the ones that are parallel to the scalp, that activity will not be picked up. So it's a pretty selective, and MEG is preferentially picking up activity from the sulci, while EEG is more picking up general pictures, gyra as well as sulci activity. So it's more full picture of the brain that you can obtain with EEG. So MEG is more selectivity, and in this way, you have a bit more information where what you're recording from, the origin of that, but it doesn't mean, unfortunately, that you have better localization capability with MEG than compared to EEG. It's a bit similar. So on the bottom, you have this figure showing activity, a dipole in the sulcus. And you see how the field lines are exiting that sulcus and exiting even the brain. So this is how it works. Yeah, on slide 27, you see more detail about the equipment. So you have liquid helium for cooling the whole thing. And yeah, it's a pretty bulky thing, as you can guess. It's not something that you can put on your head and walk around with it. So subjects are immobilized, but yeah, it's a very quiet environment. A bit like EEG, it doesn't make any noise. Yeah, on slide 28, you see a bottom view of all these electrodes. So there is a pre-mounted cap, if you like. You don't need to mount anything. You can immediately start recording. Your head is placed in that cavity, and then you start recording. So no messing up of your hair, if you like. And it's also child-friendly because you can, as I said, immediately start recording. And you can have a lot of sensors, and like 300 here. So you can have a decent coverage of the scalp with that. And yeah, and then for some reason the movie is not working, but anyway, so on the left there is a movie. For some of you that downloaded it, you can click on it. There is a movie that no, you don't change helmets for different people. Unfortunately, the equipment is expensive as it is. This is millions, right? Then if you see yeah, there's supposed to be a little movie behind the blue thingy here on slide 29. This is brain activity that is recording during the comprehension of a spoken word. Indeed, it starts with the auditory cortex, of course, where you pick up the auditory activity, and then it gets parsed and processed and eventually semantically interpreted and connected to language. So they get a spreading of activity in the brain with a sort of timeline that is referring to the different stages of processing. And on the right here you see some activity that can be recorded with the MAC arrangement. You can also record not only rhythmic activity, because this is what is shown here on the left, not only rhythmic activity, periodic activity, but you can also record event-related potential. So these characteristic deflections in the amplitude in synchrony with the onset of your stimulus. So it offers all the functionalities that we also have with EEG, but the source, the nature of the record activity is different. On slide 30, there was a bit of a comparison given there, EEG and MEG. So EEG and MEG both share the good temporal resolution. You can go to millisecond range, so you can have a lot of detail there, and so on. But it is problematic in terms of spatial resolution. You have the inverse mapping problem we talked about. It's in a sense a two-dimensional, albeit a bit bent, but okay, a two-dimensional coverage of activity that is actually three-dimensional in the brain. And then EEG has some advantages. It's pretty cheap. EEG equipment is a lot less expensive than the MEG equipment. MEG equipment is millions. EEGs is like tens of thousands of euro. You can have it of good. And you cannot do both MEG and EEG on the same time. You can't do that. You could do it not simultaneously, because the electrodes are disturbing each other, right? So electrical activity picked up and magnetic activity, the two will disturb each other. Don't forget these electrodes, EEG electrodes are metallic. They are covered, and that will disrupt the magnetic activity, the field lines, and so you can't do that. But you can do it sequentially, of course. The signal of EEG is they say large, but that's only for alpha. If you go down for event-related potentials, then it becomes in the micro range, so it could be very, very small. But anyway, so that's not the case with MEG. We are here on the order of femtotesla, 10 to the minus 15, extremely small stuff. And the signal is here distorted by the skull and the scalp. That's a known phenomenon in case of EEG, which is not the case with MEG, because it relies on a different physical principle. Spatial resolution of EEG is on the centimeter range, and for MEG it's a bit better, but essentially the localization capabilities are similar. And EEG gives you a more full picture of tangential as well as radial dipoles, while for MEG, as we said, is only within the cell's eye, so it's more restricted. With EEG, subjects can move, and you have indeed EEG equipment that is even wireless, and you can walk around, move around in the supermarket while recording EEGs. It's all possible. With the MEG, you can't do that. It's all fixed in the bunker, remember? And sensors are, in the case of EEG, directly attached to the head. You need some gel or some liquid to make it happen, and it's not the case in MEG. But on the other hand, you need to make a special laboratory and a lot of precautions. And so this is you have all of these are the basic working principles of EEG and MEG. That's something that we already discussed. Okay, slide 31. There is, of course, something new again happening, OPM. So we are fortunate to be working with the people that developed this technology. So OPM means optically pumped magnetometer. It's a special principle. It's actually a combined optical and magnetic sensor. With the previous case, we had the squid electrodes, and that's purely magnetic. Here there's a combination. And on the right side, it looks a bit like Carnival. Doesn't look very nice, but nowadays I have more compact sensors. These were the more like early stage. And what's the working principle? Look at the figure there for the panel B. There is a